Hello, I'm Miles Ratcliffe, and welcome to another edition of Organoids and a Large Portion of Chips. This is the Work.Community podcast. So I'm here on the 11th floor of Organoid Towers for a very special show focusing on the three R's. That's replacement, reduction, and refinement of animal models in research. And this episode is being kindly sponsored by Crown Bioscience, who specialise in contract research services for oncology, including, of course, 3D organoid models. Well, I'm very pleased to say that we're going to be joined by some very special 3Rs guests today. But first of all, let's have a very warm welcome back for our regular hosts, Professor Mariana Krautoff de Julio and Professor Stephen Wilson. How
Hey,
are you both?
Miles.
Hello,
Hey,
Miles.
Mariana.
Hi, Steve.
So, Mariana, what have you been up to the last few weeks?
Oh, I feel like I've been driving planes. No, I've been lots of traveling lately. I've been in many study sessions, so a lot of learning while I've been reviewing grants. So it's been really exciting times. Yeah.
So, what have you been learning, dare I ask?
Oh, so I learned a lot about how innovative some people can be in terms of looking how to monitor cancer with like hair follicles, or people have been very creative in with things, and also different ways people are also now using organoids in terms of implantations also in zebrafish. So I think the field is moving towards different things, and I like how creative people have been in this. So it's really exciting, yeah.
Oh, fantastic. I'm not sure the follicles would be much use for myself, but yes,
Well,
okay.
maybe you have some facial hair. You have facial. We start plugging that. Well, you'll never need that. And by
I'll
the way,
leave my hair growing details for another episode, but yes. Stephen, what have you been up to?
So I've been going bananas at the university with the part of my job as professor as a teacher. So the last couple of weeks I've had like maybe 50 oral exams.
50.
So yeah,
Wow.
yeah, it's been crazy, but there's some really good students out there. So I've had exams in courses in biochemistry and analytical chemistry. So I'm really happy about that. But now I'm currently in Belgium at a conference for chromatography, and just right after this talk, I'm going to be running out to hold my oral presentation here. So having a great time, lots of stuff going on.
Yeah. Hey, and listeners, if you haven't seen, Stephen's got some very cool technology in the space here. So do check him out online and in the
Yes,
podcast notes afterwards.
thank you, Miles.
There you go. There's a free plug for you. Excellent. That's what it was
It's
all
all
about.
right. Yeah, yeah,
And if anybody
that's
else
great.
has some things to advertise, feel free. That's all good. So I was saying, it's a very timely, and this episode is focusing on the three R's with the recent announcements by the FDA, NIH, and not to be outdone as well with the EU roadmap as well on this shift towards human-specific research and animal testing alternatives. So we have three very special three R's guests today. They are from the UK, first of all, Dr. Cathy Vickers, which is from the Head of Innovation at NC3Rs. From Switzerland, we have Dr. Armand Mensen from the Swiss 3R Competence Centre. And from Norway, Professor Adrian Smith, the Norwegian 3R consensus platform called Norecopa. So I thought, first of all, it'd be nice if us as podcast hosts could introduce everybody more properly. So, Mariana, would you be so kind and formally introduce Armand?
It will be my pleasure. So Armand Mensen is the scientific officer and deputy director of the Swiss 3R Competence Center. Armand received his PhD in physiology in 2012 from the University of Zurich with a variety of research fields such as sleep, consciousness, and stroke. Very, very interesting, I must say, Armand. Armand's work always focused on optimizing experimental design and developing novel statistical analytical tools. This perception, along with a strong ethical compass, brought him to join the Swiss 3R in 2018. He now manages the 3R funding scheme, the reviews on application, monitors the ongoing projects in a very strict manner, I should say, and tracks the 3R impact they might have on the research and public policy landscape. And I must add, he is always very helpful and open to chat. And so I'm really glad that you decided to join us for this chat. Thank you. Thank you very much.
Yeah, thanks very much for having me. So indeed, full disclosure, We fund one of Mariana's projects, or say we fund it. It's now over.
Yeah.
So yeah, we know each other a little bit from that.
It was a while ago, so it was a project awarded in 2019. I think it was one of the first
Yeah,
or
the
second
second year we
one.
ever did our funding scheme. So the Swiss RCC is established in 2018.
Yeah.
And then yeah, your open call 2019 project 3.
Yeah.
So
So it was a
absolutely.
very successful one. That was our two Nature communication papers came out of that project. So I would say it was very, very helpful
Well,
and
I've got to ask, what was the project very quickly?
so this was to set up the precision oncology platforms here at the University of Bern and to optimizing the GU platform for generation of organoids and to reduce the amount of animals that would be used for experimental testing. So yeah, I think we've been quite successful in doing that. So and we also generated maybe Armand doesn't know this, but we generated also a spin-off company off of this as well. So yeah,
Excellent.
kind
Wow.
So
of.
exactly what we're looking for.
Investment in action. Marvelous. Sorry, Maria, back to you.
Yeah, so of course we have plenty of questions for you here to ask, but they told me I don't have much time to just monologue with you, but here we go. Here it goes. So how can experimental design improve the use of animal models and what are the alternatives? How do you see this and what would your comment on this be?
Yeah, I mean, like your intro said, experimental design has always been a key for me, just because it's a key to any research, I think. A concept can be solid, theoretical background can be there, but if the experiment is not designed in a way to produce those meaningful results, so maybe it's unsuitable control groups or bias in the selection or insufficient power, it's still just a waste of time, energy, and resources. As we all know, money being the one of the main resources, but not the only one. And then, of course, as we move in the case of animal experimentation, it also becomes a further waste, of course, of an animal's life, or generally their life. And so it really is it's such a base factor. So, with smart experimental design, we can really limit the sample sizes, or at least be precise about the sample sizes. We can define good control groups. We can already define the proper statistics that we'll need and really while maintaining the scientific rigor that we need to actually produce meaningful results. And then even when we think about the alternative approaches, I mean, one thing is that a good experimental design in the alternative arena can give a lot of those other researchers, perhaps the ones using the animals, the confidence that yes, indeed, this is a really good research project, and I might be convinced now to move from my animal-based project over to this alternative. So it's such an initial key part of really any experiment, but especially ones to do with the 3Rs landscape.
So you would say that basically good planning and good statistics would be at the basics of the foundation of the generation of an experiment, of the planning of an animal experiment. So
Yeah, absolutely.
so moving on to something that's a little bit different, would it be maybe possible for you to explain in a few words to somebody that is outside of science, how can they actually relate to the 3R and understand this? So how would you explain this to a non-scientist?
Yeah, so I mean, the 3Rs basically as a concept started in the late 1950s. So despite that being relatively young, we've kind of iterated over a lot of definitions. And how I would denote it is that basically scientists and science at its core is about being a problem solver. So you have a particular issue at hand, and the 3R's role in that is that it tries to give a sort of heuristic or set of concepts that allows researchers to be able to solve a particular problem by asking themselves a few different questions. Can I solve this without any use of animals at all? And most problems, of course, that's quickly yes, and then we move on. But when it's not, okay, I need to use some animals. What's the fewest animals I need to be able to get reliable answers?
Mhm.
And then the last is sort of, okay, I really need these animals. The ones that I do need, how can I be responsible and make sure they're as comfortable as possible and as well taken care of as possible? So really kind of just any set of principles that allows you to go through those kind of questions and sets of questions is kind of related to the 3 R's principle.
Yeah, but don't you think that it's still a bit difficult for the public to actually understand and to be sure that we as scientists are actually using the animals appropriately and in a really necessary manner? So how can we reassure the public that it is necessary and that all we do is very well regulated?
Yeah, I mean, it's such a problem, I think, at the ground level. Like the scientists themselves are reluctant to even talk about some of these issues. Somewhat understandably, there's a lot of sort of, well, realistic risk in some of the pushback that exists. So very understandable. But in the end, really transparent reporting of research methods, kind of pre-registering your studies, having independent ethical groups kind of to review a lot of the work. These are things that are already in place for the most part. And as long as the public can have a sort of understanding that those processes are in place, that can build confidence. And then also just, or I should say, part of that understanding just relies on having really good bridges between the scientists and public so that, good communication, good scientific publication, open honesty, those are all things that can build that trust again. And I think with the trust, you can point to a lot of the systems that are already in place to make sure actually, anything that is being done is really necessary and certainly in Switzerland, very, very well regulated.
So that's really interesting because that brings me to the next question, which would be if you could just, change how society sees one thing about the research that we do with animals, what would that be? And why would you change that? Or why would you emphasize that if not changed? Or maybe it's something that's already there, right? And then we would just want to maybe give it like a spot a light on it, shine a light on it.
Right. So I think what I would want people to understand is that, along with that definition I gave about how like the 3 R's kind of should be seen, um, good animal welfare and caring about the animals and the people that work with the animals and excellent science really go hand in hand.
Mhm.
There isn't a 3 R's that limits research or wants to impose restrictions and tries to do a whole lot. It's about making what is going on like more reliable, relevant, ethical, and those things just go hand in hand. Like, I mean, there's sort of happy animals is good data, right? Or good animals, good data. And I think even though it's a short slogan, there's a lot of truth in that. And so I think if people can come to the understanding that the scientists that are doing these projects are people too, I know it sounds weird that they're also people. And for the vast majority, and certainly everyone that I've ever encountered in the field, they really deeply care about the animals they're working with. And I think if that becomes kind of understandable to the, let's say, average public person, I think we've already come a long way in building that trust.
I like that. I think that's a very interesting and a very strong point. So, which brings me then to my last question, and that would be, what does the future of animal research look like in Switzerland and maybe globally? I mean, a bit of an ominous question, but what do you think? You know?
Yeah, well, I mean, Switzerland, as much as we like to think of us in a certain isolated way, we're certainly not isolated. I hope Switzerland is, and I hope it continues to be at the forefront of a lot of this research. I think on a practical sense, obviously, it involves more sophistication in models, organ on a chip systems, organoids. AI is probably going to become a big thing, and that's generally,
Mhm.
obviously. And I hope that also whatever remaining animal research there is, it just becomes more precise through kind of these experimental designs, statistical ideas, and more responsible as we continue to grow our knowledge about kind of animal welfare, how to measure animal welfare, and also like what is the animal's internal experience. And we can sort of guide a little bit of that. And so hopefully there still are these different bridges, all encompassed within the 3Rs principle that continue to kind of grow and define. I think as most science is, it'll be slow and steady, and it may take some time. But I definitely think even in the 6, 7 years that I've been part of the 3RCC, we've seen a lot of that growth already happening.
Well, that's great. So I hope that that group keeps on going in the right direction. I'm going to hand over the mic to Steve for
Yeah.
the speaker. Well,
So-
thank you very much, Armand.
Sure.
Thanks a lot. And now we're going to be talking with Adrian. So Adrian Smith is a British veterinarian who graduated from Cambridge in 1979 and emigrated to Norway in 1980. And he held a chair in laboratory animal science at the Norwegian School of Veterinary Science from 88 until 2011, and has arranged over 50 courses for researchers and technicians, and been the secretary of Norecopa, which is Norway's consensus platform for the 3Rs, which are replacement, reduction, and refinement of animal research, as we all know, since it was established in 2007. And he's had a special interest in 3R resources for many years, which I have also had the pleasure of really being able to use in many different situations, also when preparing for talks and so forth. And he's co-authored several databases in this area, and is the lead author of the prepared guidelines for planning animal studies. So Adrian, it's really nice to have you here. And I also know that in Norway, you're also well known for trying to influence the policies in Norway. Could you tell a little bit about what is like the political landscape in Norway when it comes to 3R initiatives? Can you tell a little about that?
Yeah, thanks very much for inviting me. We might just add one more disclosure. That is that I got to know you through Norecopa's 3R Prize, which we award every year for a person or group of people who have helped to advance significantly the 3Rs. And you had 3 students who were nominated last year, and although they didn't win last year, I encouraged them to nominate themselves again this year, and this year they won the prize. So congratulations with that. And that was for work on organoids.
Yeah, that's really cool. Thank you so much
That's
for
one of
that.
the ways in which we try and spread information about the 3Rs to the general public and the politicians as well, by showing that good work is being done in Norway to promote this concept. So that was maybe an initial answer to your question.
Are politicians in Norway hard to convince about 3Rs?
Well, there's a real mystery going on here. I mean, it's a total no-brainer to be for alternatives to animal experiments. I mean, which politician would be against alternatives to animal experiments? I mean, obviously everybody is for it, but going from that to getting them to pass legislation or establish a proper 3R centre has been much more of an uphill battle. And my impression is that every time we see a chance to have a really good debate about animal research, then another group of animals hits the headlines. Say it's tragedies on the pig farms, or the way we keep poultry, or whether we should forbid fur farming, this sort of thing. These other scenarios, which the general public seem to relate more closely to, have sort of taken over and swamped our debate. And therein lies the dilemma from my point of view. Norecopa, which is trying to promote the 3Rs, is primarily targeting the scientists. And so we don't want to go out in the mass media with sort of blood dripping photos of lab animals that are suffering, because then it would alienate the group of people who we're most interested in talking to. So we keep a fairly low profile, and now at any rate in 2025, most of the animal welfare organizations are actually basically doing the same. Then we don't see the same level of antagonism in social media and the papers that we saw, say, 20 years ago. So as a result, I think the general public seem to be sort of thinking, well, we rely on the fact that scientists and people like Norecopa and the local animal welfare bodies are taking care of this. We don't really want to know more about animal research. And this, I think, probably influences the politicians, because there aren't a lot of people lobbying outside the parliament to abolish animal research. And the main problem from our point of view has been that we haven't managed to get sufficient majorities in Parliament for big changes.
Yeah.
Norecopa is a small organization. We would like to have a much bigger 3R centre. We hoped that that would be proposed in a governmental report that was published just before Christmas on animal welfare. But the reports just concluded that the government would evaluate the need for a centre and how a centre could use existing resources like Norecopa. So
Yeah.
we are now in a sort of process where we're waiting for this evaluation, and it's nice to see that Norecopa itself is clearly appreciated by the politicians, but we'd like to go one step further and have a much bigger 3R centre, so we could relate more to the 80 lab animal facilities we have around Norway. There are a lot of them, and it's a big country.
It's really interesting, Adrian, and I'm really thinking about the point that you're making, that, I mean, of course, animal research there's a lot of problematic aspects with it. Of course, as we've discussed previously, I think animal research has been a clear necessity, and will have a very strong role in research going forward. But as we're talking about here, one wants to be able to do it smarter, or make it more alternative models, like you say, organoids and organoid chips. But it seems like you are, it can get in the way if people are getting too dramatic in their way of communicating what is going on and so forth. I was just wondering, maybe Armand also, would you agree with this notion, for example, that the way of communicating the wish to reduce animal experiments would you agree that the way is to do it in a more of a scientific way than in a more of a dare I call it, an emotional way? How does that sound, Armand, would you say?
Yeah, no, I think that has to be the way to do it too. And otherwise you get like this defensive thing coming into it, and you defend your project, or you defend your research career or something. I think if you feel that that's under attack, then you're unlikely to even hear the next sentence, which is, how and why and what resources it take. I mean, it would just probably be a block from right there. And I think there's the majority of arguments that I think one could have when there are realistic alternatives are perfectly based within science, or even things, if you're a politician, perhaps your major interest might be cost effectiveness. And there are great arguments within those frames too. So I think you do have to adapt your message a little bit. I think the scientific route is almost certainly going to be your best bet in most ways. But yeah, it depends on your audience. Get to know them, what they care about, what type of politician that they are, in terms of where they are in terms of, the Ministry of Finance. Well, then you may be talking in a different way than, the Ministry of Agriculture might be interested in.
Yeah, sure. Yeah.
Could
Well,
I
listen, Adrian, yeah,
Yeah.
shoot in, Adrian.
I've just come back from the big European Lab Animal Science Congress, PhilARSA, which is was held in Athens for a whole week. We were over 2,000 people there, and it's very clear that the science of laboratory animal science, the field has really advanced, and we have a lot of good information there. But we are not as good at communicating with the other scientific fields. I would like to see far more outreach to the neuroscientists, the physiologists, the toxicologists, the pharmacologists, etc. I think that's really what we need now, far more dialogue between these groups. And as Armand mentioned, I think some of these scientists quickly feel threatened if we mention the three R's. They assume we're trying to replace an animal model which they maybe spent 30 years building and humanizing and writing papers about. And who are we to come and say that, scrap all that and start using organoids instead? So, but at the same time, I think there is still probably a lot of opportunities to improve and refine the animal work that has been doing, is being done. And I've noticed the few times I've been at these other congresses when I'm looking at posters describing, say, the anesthesia analgesia that's being used, that we could have been there and given them some better ways of doing things. And I think we need far more of this dialogue because animals research isn't going to disappear in the next 5 minutes, and we need a lot more guidelines, both species specific and situation specific guidelines to improve what is being done. That was why Norecopa spent so much time developing the prepare guidelines for planning animal research. They were initially based on all our experiences talking with scientists, running courses for them, managing accredited facilities, and realizing all the practical details that can ruin an experiment which otherwise has been planned well scientifically. And I don't think all scientists are aware of all these small practical details. They assume the animal facility is dealing with it, and we need to engage them from day one of planning to make sure they have sufficient dialogue with the facility to plan good research, and that's where prepare comes in. And then at the other end of the research path, then we have reporting guidelines, which Cathy will no doubt tell us more about. So we need to take care of them from day one right until they submit their manuscripts.
Yeah, well, maybe that's a nice segue to Miles. Maybe we'll be hearing more from Armand and Adrian also in the last roundtable part of this. But Miles, maybe you would like to introduce Cathy to us.
Yeah, absolutely. Thank you so much, both. So Cathy Vickers is at Head of Innovation at the UK National Centre for the 3Rs. Cathy received a PhD in molecular neurophysiology from Edinburgh University and has spent time working in Japan, the US, the UK, in both academia and industry before joining the NC3Rs itself. She leads the Crackit Open Innovation Programme, which is funding collaborations between academia, SME, so small, medium business enterprise sector, and industry scientists. And the purpose is to develop commercially available 3Rs technologies that can be used in the real world across sectors to deliver better science, improve business processes, alongside having, real world impacts on the 3Rs. So thank you, Cathy. Welcome again.
Thank you.
And
nice to be here.
pleasure. And so I think, within the UK and the rest of the world, the NC3Rs is very well known within the research. And it's about as old as I am. So over 50 years ago, I think it's been going. But just to tell me a little bit more about those important links to industry in the Crackit programme and how the industry is involved in these replacement efforts, and maybe you have some favourite examples.
Thank you. Yes, Miles. I think industry is very interested in replacement, and by industry we mean across all sectors. So that covers pharmaceuticals, chemicals, agrochems. We know the cosmetic industry already do not use animals in their regulatory testing, but also CROs. the contract research organisations that support these large industries, and also a very fast-growing small business sector that is growing, spinning out from universities and providing some of these real key replacement tools and technologies. And I think industry's interest has always been there, because obviously we've talked about it, you know, I think everybody has that ethical driver to implement the 3 Rs. And if we're talking about replacement specifically, you know, I think all industries have that ethical driver to not use animals where they can avoid it. But I think what's happened recently is, I think probably for the first time, we are now in a position with iPS cells, with organoids, organoid chips, in silico modeling, we can actually study human biology and disease using human biology as the model, as opposed to an animal model. And I think many people are aware, especially for things like pharmaceuticals, that failure rate or attrition of drugs failing to get to the clinic, so failing to be prescribed and available for use in humans, because the data that was generated during their testing in animals does not translate to what happens in the human. So it might be that it appeared to be safe in the animals, and then there's an unexpected toxicity in the humans, or it appeared to work in animals and doesn't seem to work as well in humans. And I think that's been a huge barrier to some of, a huge cause of attrition to especially the pharmaceutical industry, but also to other industries when they're talking about trying to ascertain whether their products are safe. And as I was mentioning, now we've got this huge advances in biology and cells and being able to recapitulate some of human physiology better in vitro, that really does open up the opportunity to study, develop and test new medicines, for example, using human biology rather than animals. And I think, I just wanted to highlight that all of these products, whether they come from pharmaceutical, chemical industries, etc., they all have to be approved by regulators. And these are government agencies that look at all the data that's been provided by the company to make a decision whether the, let's say, drug in this case is effective and it's safe. And I think that is where animal studies are still required. So a lot of pharma, I'm talking pharma specifically, will use a lot of in vitro complex models, human models. They're still submitting animal data for regulatory approval. But we know that the regulators are also excited about the potential of replacement with the recent FDA announcement of their roadmap, their plan to phase out animal tests for safety as a priority, preclinical safety studies. EMA, we know we've got the 3 Rs working party in the European Medicines Agency. So there's a lot of momentum, a lot of appetite. But I think what that generates is a huge amount of interest, a huge amount of momentum, and a huge amount of promise. But I think realistically, it's about setting expectations over time. When can we get to the point where we can say you no longer, for example, need to use animals for regulatory safety studies? There are instances now where people have put submissions in for approvals, whether it's a new indication for a drug or whether it has been supported by in vitro studies, but it's not the norm at the moment. But I think there's a huge amount of interest and engagement because of the human relevance, potentially cost and time efficiencies as well. I think it was Armand that was mentioning that, you know, if you can avoid doing a 9-month study in an animal because you've got advanced in vitro and in silico models that you can run from your laptop or from your bench side, that obviously speaks to lowering costs and time as well.
And in terms of those projects which you're funding, how much of the innovation is maybe coming from scientists using animal models at the moment, or is it more people coming from outside thinking maybe how can we change this? Is it a bit of both, or?
So the way we frame our CRAFIC challenges normally is we're working with industry scientists, and they are either currently using an in vivo model to address a specific particular scientific question, let's say, and the animal models are potentially, they're very burdensome. So perhaps the animals are going to go long studies. There could be some pain involved, for example, or it could be that they're not just producing human relevant or interesting or relevant data. They could be taking a long time. And then the scientists who come to us from these industry sectors are also aware of what's emerging from the academic and SME research sectors. You know, iPS cells are being Organoids are starting to show how you can address this problem with microfluidics and engineering. You can create these complex models that, with a little bit of extra funding, time and challenge, maybe could be used to replace some of the animal studies that are currently going on in R&D. And that's how they're set. Some of our challenges are for full replacement, an in vitro model to look at nephrotoxicity to replace the use of the rodent. Some of ours are looking at better, just more human predictive biology, the development of, but we also look at in vivo challenges as well, where perhaps there's the opportunity to improve the welfare, so refine the use of animals that are being used in these studies, reduce the numbers of them that are being used. But always, and again, I think Armand and Adrian were mentioning this, it's science-led and evidence-based. We're pushing the science forward by integrating all of this new knowledge and expertise within the projects that we fund.
And do you have if you could single out a couple of examples, or maybe just one, which you think
Yeah, so
is
one
a
of the
really
ones
good
we have running
example?
at the moment is we are funding the development of a virtual dog. So
Oh, wow.
dogs, so when their medicine is going to be approved, or when you're testing the toxicity of a new medicine, and without getting too technical, this is mainly in small molecules we're looking at here, you have to do toxicity in a rodent and a non-rodent species. The rodent is normally the rat, and the non-rodent species is normally the dog. And you will do, again, it varies, we can't go into regulatory guidelines, you'll do a short-term and a longer-term study in both species. We have worked through some of our other programs at NC3Rs to identify that actually, retrospectively, looking at data and decisions that have previously been made, we analysed a data set that showed that the use of the chronic study did not change a decision that would have been made based solely on the short-term study. So what they're saying is the regulators require to do a short-term and a long-term study in the dog, and retrospective analysis suggested that there were times and opportunities that the long-term study in the dog didn't alter the decision around the safety or the information around the molecule. So what we're doing, so getting regulatory requirements to shift to say that you don't need a chronic toxicity in the dog would take a very long time and needs a lot of evidence. And part of that evidence we're trying to generate through the creation of this virtual dog. So this is trying to use advanced in silico modeling and machine learning to create a suite of virtual dog organs so that you could make a decision that you would normally make following a chronic toxicological study using a dog, using our in silico model. Now, it's very ambitious. I'm not sure the dog will have a waggy tail and ears, but what we're doing is
And how many different parties are involved in this?
So we have 7 pharmaceutical companies. We collaborated with the recent IMI project eTranssafe, and we're working with a German SME, ESQ Labs, collaborating with Fraunhofer to deliver the work. This is quite a big project. It's got quite a way to go. It's a big challenge, but again, just really excited to say that awareness of the capabilities that are coming out in the scientific research phase and matching them up with some big needs from industry can actually deliver really exciting and innovative tools that can really have an impact. And we've got a similar bigger challenge is looking at organ on a chips. So looking at connecting organ on a chips, or what they are, we know this, but Organoids, organ on a chips podcast. But what we're looking at is reducing some of the barriers for their sort of rapid uptake and integration into industrial workflows by getting that confidence, making them more automated. So we've got a big challenge called SensorChip that's looking to develop with real-time multi-parametric monitoring of these connected organs on chips, and also combine them with some advanced data interrogation and machine learning so that you can get that robustness, reproducibility and reliability, which is perhaps lacking from some current organ on a chip systems because of their complexity. And you need to be pretty skilled at doing your organ on a chip experiment, and reducing some of the barriers to their incorporation into sort of R&D, but also looking to say, can we get as much high quality, reproducible scientific knowledge out of these chips as we can by collecting all the information we can and doing some really thorough analysis on them.
Fantastic. That's really exciting. And for everybody listening, we will be putting some links at the bottom of the podcast as well on some of these crack it and other challenges. So thank you so much, Cathy. That's wonderful. We're going to move on now, have a bit of a round table discussion. So we're going to have a few questions going to pop out there. And I think one of the main ones is, what are the biggest challenges, but also the opportunities, Armand, Adrian, and Cathy, that the three R's face today? So really the challenges and opportunities which are faced by everybody. Maybe Armand?
Sure. Although I'd love to defer to my better knowing colleagues, but I think certainly one of the biggest challenges we face actually in the 3RCC at least, but I'd be very interested to know if my colleagues have the same, which is simply a lot of good science already exists. So there's a lot of really good like individual kind of papers that are out there. You can look for them on a number of topics, whether it's organoids or organ on a chips. A lot of the really good basis science has been done. And then the paper kind of sits there by itself a little bit. And we don't have this widespread outreach by the scientists. Well, one could ask, is it really their job to do this? They've done so much of the work already. But to get that to, neighboring systems, maybe a colleague of theirs uses it, maybe someone picked it up at a conference somewhere, but it's not as widespread as it should be, or could be. So there's a lot of this low-hanging implementation fruit that I think can be had. So that's the challenge. But because it's this low-hanging fruit, we also see it as this opportunity where, man, the 5 years of hard work and the publications that you have to get through the system, that part's over. And there's probably just these little niche things that maybe aren't as convincing. Perhaps that's understanding. Perhaps that's, maybe detailed costs or marketing or things like this that aren't there yet. But for us anyway, implementation of already established, scientifically established methods is really one of the biggest hurdles and opportunities for us.
So that's really sort of a communication and engagement point of view.
Yeah, there may also be sort of, minor research questions that prevent kind of a full uptake, like, oh, will that work in this particular thing? But by and large, I think it is a little bit of just communication and understanding.
I mean, do you think these questions go on directly to the author, or do you think somebody reads a paper and then it gets lost and they move on to something else? Or
I think there's a lot of reasons, and I'm happy to hear from Cathy what kind of things she most often comes across in this space.
Yeah, so
thanks,
maybe
Noel. Thanks,
Cathy,
Miles. Oh.
maybe Cathy first, and then Adrian has a comment after that. Okay.
Okay, I just wanted to add, we set ourselves quite a task. We have 3 ambitions: replacement, reduction, and refinement. And we want to implement them across the whole of basic life sciences research, pharmaceutical R&D, agrochemical, petrochemical, veterinary medicine, and so it's huge, right? The whole fields and the whole areas of life science research is huge. And I completely agree with Armand. You kind of sometimes can feel frustrated that, these models, alternatives, or refined models or experimental that is out there, it should be, and they are easily accessible. It should be more, whether it's communication, whether it's better dissemination of these approaches, but that encouragement to drive or incentivize in vivo researchers to move away from that. But I think we have to be a little kind to ourselves in realizing the size of what we're trying to change, just in the disciplines and the research areas as well. But just before I hand over to Adrian, I also wanted to say it's about expectations as well. And I sort of alluded to some of that when I was talking about some of the regulatory sides. Organ on chips and organoids, they're really exciting. Of course they are. We can do things, as I say, model human biology like we've never modeled it before, but we can't replicate a human in a dish at the moment. We can't replicate where a drug travels in one heartbeat, its exposure, its metabolism to all the different organs and all the different. And so I think sometimes the expectations can be too high from these technologies right now. They are so good at doing what they're designed to do, but the expectation that they could solve every scientific question or deliver data against every hypothesis right now is not there yet. The important thing is to make sure they're implemented in replacing animals right now where they are able to do so, which is across a lot of disciplines and types of research. and then building that confidence in them so they can be further developed and combined with other approaches to then tackle some of the larger whole body, if you like, or systemic physiological questions in the future. But sorry, I'll be quiet, hand over to Adrian now.
Yeah, hi, thanks, Cathy. That's great. This is a fascinating area. It was actually something that Russell and Birch were conscious of right back in the mid 1950s when they wrote a letter of introduction to scientists who they were going to interview. And they were asked to take out the word alternative from the letter because they were afraid that the UFO was afraid that the scientists would react negatively to that. And we talk often about two different types of alternative, NATs and NAMs. NATs being the non-animal technology, and NAMs being new approach methodologies. And the definitions are debatable, but the point is that these new approach methodologies, they are often not replacements to animal research at all. And people may not even be aware they're using or developing a technique that might replace animal research. And this is yet another reason why I think we need to get out of our comfort zones and visit the other scientific societies, go to their meetings, because they don't seem particularly inclined to come to lab animal science meetings. We need to invite ourselves to them and have more dialogue to break down these barriers and show that we're all working towards the same scientific aims, and there may be alternative methods that they haven't thought about, not as a threat to their work, but as a way of advancing the science. And so I'm really a fan of more respectful dialogue between the different scientific disciplines, getting out of our comfort zone, going to those who maybe don't know as much about lab animal science as the specialists in that field. And that's one of the reasons why Norecopa spends a lot of time building up a comprehensive website on 3R alternatives so that the resources are easily available for people who are curious about that. But there's no substitute, in my opinion, to going to their other meetings and having these informal discussions about the way ahead to remove the sense that we're threatening scientists who have been using animal models for years.
Yeah, I think more sort of like knowledge transfers between labs that are planned right from the beginning. We're going to do this, and then in stage 2, we've already organized that, an expert from here and here is going to visit our lab, and also the trainees are going to visit these other labs, and have that integrated within the time scope of the scientific project itself would be, I mean, usually advantageous. Although time, who has the time for these things? But still, I think that's the communication, knowledge transfer should be kind of baked into the scientific program, along with the 100 other things.
I just wanted to add something. I mean, I think this is a really great topic and a really great conversation. But I think that we also have to focus this or look at it from the scientist point of view, right? Which is a very different way of looking at the animal research. And what I'm trying to say is that what counts for us is also of course, the kind of output and the kind of impact our output has, right? This is the meter by which we as scientists get judged, right? And we as scientists that review our own works, right, as peer reviewers, we are the worst in this aspect because what we do is that we set the bar at a certain level, right? And then we say, okay, you've done this experiment, you've used 25 different kinds of ex vivo models, and you've done maybe the zebrafish, which is not really an animal for unknown reasons, but you haven't done the experiment in the mouse, for example. And therefore, your impact is, yeah, it's okay, but you have to do that in vivo experiment. So, and that's, I think, this is part of the problem. I mean, we are part of the problem, meaning that we should also be open to see the approach in a different way and to see that indeed maybe the replacement of the animal model with the organ on chip model can be or could be something there. But I don't think we have the data for that yet. Cathy, go right
Yeah,
ahead.
Mariana, just commenting on that, I think that's absolutely true, because a researcher is pursuing a deeper understanding of their area of research, and they will use whatever, not being so the models that will provide them with that information, whether that be an in vitro, in silico, an animal model, a lower organism, etc., etc. But I did want to add, and I think that is true, and also you get that interaction sometimes with publishers. That looks great, but have you done it in an animal model? When you give us that data, you can publish it. That's another part of the struggle as well. But I think I would say, so I have been NC3Rs for nearly 13 years now. And I think it's changed even in that time. You will always have researchers who are wedded to their animal models for whatever reasons, and may be hard to entice to sort of collaborate and explore. But I think, some of these, especially as, again, going back to the huge acceleration in knowledge and technology that's been sort of unleashed through microfluidics, organoids, iPS cells, the appetite's there already now for researchers no longer thinking in vitro is second best, but actually considering some of these alternatives as being what they've been looking for to enable that study.
Oh, don't get me wrong. I would love that would actually be the new reality. One of the things that we do here at the University of Bern is that we run co-clinical trials with organoids, right?
Yeah, fantastic.
Exactly. So what we are trying to do is to run phase 2 trials or even phase 3 trials that don't require, that can use actually the predicting skill of the organoid and the AI that we are developing to bring this to another level, so that we're going to bypass the whole concept of maybe, having to do repetitive biopsies on humans even, and not use the animal work and have we have one of the Europeans' largest repository of live organoids. So, and we share these models with people. But what I'm trying to say is that I realize that we as scientists are almost the worst in judging ourselves. That's the kind of, and I do agree that it is changing, but we have been faced with so my research has been faced, and in terms of you can't get into that journal until you do that animal experiment, and the answer to me was, well, we're not going to publish there because I don't want to do the animal experiment. So that's the answer that you should give.
Yeah.
Right.
Yeah, you can probably only afford to do so with a certain level already of recognition. So yeah, I hear that. It's unfortunate, and some effort probably should be made everywhere to just align these incentives, right? So align the incentives to do really, really good work that's 3R's principle backed, and
Oh, we lost your sound a little bit there, Armand.
I can just sort of add to that while Armand's getting back online. And I think some of what Marianne was saying was alluding to some of that training. If you've been trained throughout your career in vivo skills, it's quite a big step to refurbish your lab with tissue incubators, cell incubators, learn all the skills. And so I think support to help facilitate some of that transition, not only in design, but also in the skills and training. Sorry, Armand, I don't know if you're back online now.
I don't know.
Yes, you are.
I'll hand back to you.
There you are.
Well done. No, it was really, I think the end there that was already said by you, which was just making sure that we have those incentives, and hopefully like things like the funding programs crack it already make something like that and incentivized. We have some parallel funding schemes within the 3RCC that try to incentivize that, but I'm not sure where the NC3Rs or the kind of incentives that you are able to give align with the national incentives. But certainly for the 3RCC, we're a drop in the funding bucket for scientists, unfortunately. And yet our demand is still pretty high. But but certainly some thought and planning needs to go along in that direction for incentivizing the right ways to do things.
But what about if, you three authorities that are here in the 3R world would get together and think about a way that we could work together as scientists, like find a funding scheme that, for example, would allow Steve and myself to put a grant together. We come from backgrounds that are completely different. But working together could be something that we could bring. And what I'm saying is that the funding schemes that we have are nationwide, so they're focused on what we do. And I'm not saying that we don't have the innovation and the technology and the scientists and the brains in the individual countries, but doing science together across borders is much more fun and could open doors that are not only within our own country. And I'm saying this because the grand challenges are due tomorrow. So that's why.
Very topical for you.
Yeah.
But okay, so that's
I think
that's the idea. I mean, the fun I have had in the last few weeks in brainstorming with all these people across the globe on how we can work together, how we can make the new organ on chip, how we can reduce the animal work. That's just what we do. That's fun and stimulates our intellectual, I would say, capacities to new challenges, I think. Yeah.
I agree also. I think that if, when we team up and I think that at least in natural sciences, we're good at making teams, and big and international teams can make such an impact. And we can see this both in clinical research, can be in improving analytical technologies and so forth. And I think that also seeing how European 3R centers and persons like yourself collaborating will be a very important part of that puzzle. So that's really cool.
Yeah, I think there's some great collaborations already. I know Adrian and well, Norecopa and the 3RCC directly collaborated on some data analysis things, and we're within the Acopa network as well by having sufficient stakeholders. We're in a part of. I think the challenge comes in when it's about sharing money. And not only for the obvious reasons, but I think about how we are funded and then the mandates that were put under by those funders as well. But certainly that addresses some of the implementation kind of hurdles that we have. If already a whole bunch of research groups are within the project, then clearly they will already be comfortable with the implementation. And the more widespread those things are, then that's a sort of multiple seeds approach that can actually grow from, rather than the single laboratory approach. And we know that generally collaborations do produce better science. They kind of hold each other accountable through a bit of competition and a bit of kind of different perspectives of thinking and reading the same material. So we're definitely for it, and certainly with our funding schemes, we definitely encourage it, in as much as the funding even allows for some of that.
I'd like also just to ask, also related to collaborations, and Adrian, you were touching upon reaching out to other research areas and other resources. I was just curious because I've seen that in humanities in Norway, there's been some it's starting to be some more focus on alternatives to animal research. I saw that the Norwegian research funding gave some researchers in humanities a grant on this recently. I was curious, Adrian, do you think that the involvement of also psychology and humanities would be very relevant for natural scientists, or do you think that that is still a bit of a parallel run, so to speak?
Yeah, I think I prefer to see priority given to more dialogue between the sciences, as I mentioned, that neuroscience not least, who have these enormous congresses where many animal models are presented. physiologists, etc. And another thing I wanted to mention in that connection is, which I think is probably on your notepad of things to talk about, is collaboration between the European 3R centres and what they can do. We have a cost action called Improve that has worked to establish a network of European 3R centres. We have about 30 of them now. We have something called EU 3R Net, which has had several meetings where we've spent most of the time so far just getting to know each other, but certainly planning the way ahead to collaborate so we can be better at spreading information. And then to scientists who maybe don't understand the full ramifications of the 3Rs. So I personally would like to concentrate on dialogue with those who are using animals in products like in the neurosciences.
I see.
Yeah,
Thank you.
I think there's because of the low-hanging fruit that I mentioned before, like there is a lot that can just be done when you think about the basics. Like a lot of it isn't, oh, we're having to fine-tune some of the ideas and concepts and reach out. So there's a lot of groundwork to be had. So, I agree with Adrian there. I think there is, and there's a few projects going on in Switzerland that maybe I can provide a link to later, but which are humanities-based projects. They're really looking, it's a bit of change psychology. So how do you motivate people to change their minds? And that sort of area of the humanities, I think is quite relevant here. Again, with this implementation problem, and just figuring out what it takes for, let's say, an older, established research group to go, you know what, it's time for us to move to a different path. And I think there's lessons that we can learn in the humanities.
Do you think, Armand, as in your background in psychology, do you find that you can see things in a somewhat different angle than one who's been working his or her whole life strictly in, for example, cell biology?
I don't know if the background discipline gives you that. I think there's just sort of, and for very good reasons, just different sort of personality types and different priorities that people within the research groups have. And perhaps, again, it might be an incentive question where the sort of people that don't mind doing kind of the low level grunt work and planning work, they're not necessarily the type of people that then become leaders of the group and determine the research strategies. So I think there may just be more low level things going on there than let's say academic background or career ambitions and things like that.
Interesting. Well, thank you, everybody. We've been here for about an hour, and the conversations, I think, could keep going for several hours, which is that's what it's all about. And the communication is key. And that's what we're trying to do at Work Community, is to make information accessible to everybody. You can't have everything, but I think what's always important is to have relevant examples and success stories for people to hear. In this modern world of TikToking, social media, and all the rest of it, how do you disseminate the noise? It's trying to reach people quickly and effectively before going into the detail. And that's also part of the events like Word put together, which reminds me, actually, a long way off yet, but on the 4th of December, we're having an organoid and chip Christmas party with Queen Mary University London at the Centre for Predictive In Vitro Models with Martin Knight and Hazel Screen and Stefan Verbruggen there. We've got speakers from Pharma, AstraZeneca, GSK, Cosign, and lots of academia as well. And the chance to have some proper conversations as well for several hours. And there might be some fizz, lots of mince pies, well, that sort of thing as well. So lots of events coming up. But I'd just like to say, I think we're going to have to wrap it up now, but just thank you so much to our special guest today. Thank you to my regular hosts as well, to Steve and to Mariana, Thank you, everybody, for joining for another episode of Organoids and a Large Portion of Chips. And we hope to see you all again soon. Take care. Bye-bye.
Thank you, everyone.
Bye.
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 Thanks for having us.